tissue. Unfortunately, you know, the blocks and the seminars must be very concise. So we will not be able to talk about all important things which concern the lymphoid tissue, its organization and function. We will focus only on the organization and function. So let me go to my screen now. Okay. Yes. Do you see my screen? Yes. Okay, thank you very much. So we go to the presentation. Well, okay. Can you hear it? Can you hear me? And do you see what is on the screen?
Yes.
Okay, thank you for your feedback. Well, let's start. So we are talking about the lymphoid tissue, and the first important thing I would like to remind you is that the lymphoid tissue as such is a part of some quite big thing which we call the immune system. The immune system, just in brief, is characterized by the cells and tissues which actively participate in the immune response. So this will include what we call lymphoid tissue, as well as the cells which are found in the blood, white blood cells namely. And what is also important to stress that the immune system also includes many other tissues, let's say all other tissues, which have cells which may be included into the what is called immune response, maybe except the central nervous system and the eye and the testes. Well, the cells of the immune system, there are many of them, and these which are listed here on the screen, these are the cells which we are used to call professional immune cells. This will include lymphocytes. Among the lymphocytes, we can distinguish T lymphocytes, B lymphocytes, as well as NK cells. T lymphocytes and B lymphocytes are responsible for something which has formerly been called acquired immunity. Now we call it adaptive immunity, whereas NK cells belong to the system which does not belong on this very special reactions to be activated. So they belong to what we call innate immune system. The cells of the immune system, except lymphocytes, also include the cells which arise from the monocytes. The monocytes, as you remember, are released from the bone marrow. They go to the circulation, and in the circulation we call them monocytes. When they go to the tissue, they undergo the process of differentiation and may change into the macrophages or dendritic cells. Finally, we have also granulocytes, 3 types of granulocytes, as you remember from our blood class. They are present mostly in the blood, but they have a possibility to go to the tissues where they participated, usually or mostly in responses against bacteria. Finally, the immune system also includes mast cells. The mast cells, as you know, are present in the different connective tissues, where they play a role in initiation of the local immune response by release of many mediators like histamine, for instance, but not only histamine, some cytokines as well as. You know, in most of the histological handbooks, The mast cells are included as the cells of the connective tissue. But as a matter of fact, they are the true cells of the immune system. Normally in the connective tissues, they arrive from the outside, so they are immigrating cells. Let me also remind that all cells of the immune system originate from the bone marrow. They are bone marrow-born cells. Well, how what we call the lymphoid system is organized. You know, the role of the immune system, it is to defend our organism against pathogens, external and internal. Therefore, you see, it is not surprising that the immune system has an organization which somehow resembles the organization system of the armies of the countries, which are devoted to defend the country from the external enemies. If you want to organize an army, you have first to take care about proper, education of the soldiers. So every country has the military academy, like the famous West Point in the United States, and also is organized into some units. These units usually they stay in some special garrisons or in special posts. If we follow this way of organization, we'll find the striking similarities between our immune system and the organization of the army in the country. Because in our organism, we have something we call primary lymphoid organ. These primary lymphoid organs include the thymus and the bone marrow. As you know, the bone marrow, it is to produce the lymphatic cells, so it is like a factory which produces the soldiers. But these soldiers who leave the factory are not trained as yet. Therefore, they need some training, especially the cells of the adaptive immune system. Therefore, after leaving the bone marrow, the precursors of T cells via circulation reach the thymus. And the thymus is such a, school for the T cells. B cells, they have there is no necessity for them to leave the bone marrow, because the bone marrow also plays a role of the military school for B cells. So the B cells stay in the bone marrow and undergo military education at site, let's say, whereas the T cells go via circulation to the thymus. So we have primary lymphoid organs which serve as military schools. When educated, the cells from the primary lymphoid organs, they leave these organs and go to serve outside. And they settle down in what we generally call the secondary lymphoid organ. The secondary lymphoid organs are divided into two major classes. First, there are encapsulated lymphoid organ and unencapsulated lymphoid organ. Encapsulated lymphoid organ, these are organs which are surrounded by the connective tissue, which forms a kind of a capsule inside. And this group of organs include lymph nodes and spleen. What is important is that they are separated from the rest of the tissues, rest of the organs by the capsule. So they indeed form the environment which is very specific. Hello? Once more, please, because I barely hear you. Hello? Can you hear me? I have no feedback. Can you hear me? Hello? Can you hear me?
Yes.
Okay, because I have heard some voice on the back. So okay, so let's continue. Okay, all right. We go back. We go to the presentation. Well, so the as coming back, okay, these encapsulated organs, they provide some very specific environment. But some immune cells form aggregations in the connective tissue, especially of the mucous membranes. And these unencapsulated aggregations, which are relatively small compared to the encapsulated organs, we call mucosa-associated lymphoid tissue. That means the tissue, the lymphoid cells, which are associated with mucous membranes. So as you see on this slide, the encapsulated organs, they play a kind of a filter, especially to the lymph nodes, to the antigens in tissues, and spleen to the antigens found in the blood. Mold is responsive to the antigens which reach the mucosal surfaces, usually in the alimentary, respiratory, or urogenital tract. Well, the lymphoid organs are distributed among the whole body, along the whole body, except the brain and the eye, and also the masculine testes. So this is a schematic presentation. Where can we find them? As you see, the lymph nodes, which are shown here, that are present in many parts. In green, there is a mucosa-associated lymphoid tissue. They are at the border in the organs of the alimentary, as I mentioned, respiratory tract and urogenital. So it's generally they cover the whole body. Well, so let's tell a few things about the thymus. The thymus plays a very similar role as bone marrow for B cells. First of all, the thymus is responsible for T cell maturation, as well as the phenomena which we call negative selection of T cells and positive selection of T cells. The thymus is a place where these cells are educated. It means some cells, especially autoreactive cells, are eliminated by negative selection. And the cells which are the best suited for the immune response in the context of what is called major histocompatibility antigens, we call positive selections. We have no time because it's a special topic for the completely another course. So the only thing is that please, ask your teachers about some specific explanations. We have no too much time to follow this subject. Well, so the thymus, it started activity during the fetal period. And what we can see very distinctly, this is a beautiful picture of such a fetal thymus. This is a cortical and medullary part. You see the difference between the cortical part and the middle part. Medullary part is very apparent, so it don't need any special illustration about that. The part which is responsible for multiplication of the cells their maturation. This is the cortical part, whereas the medulla part serves first of all for the supply of the thymus with the new incoming lymphocytes. And of course, this is also the place where these lymphocytes leave the thymus and go to the periphery. Every organ is based on the special kind of a scaffold, which is generally composed of a connective tissue. This is also true for the thymus, but what is important to stress that the scaffold in the thymus is also composed of very specific epithelial cells of the endodermal origin, which are responsible for education, let's say, of the T cells. So the connective tissue scaffold is responsible for the keeping the general architecture of the organ, and also make a support for the blood vessels, whereas the cells which are responsible for maturation of the incoming lymphocytes, these are epithelial cells. These epithelial cells are responsible for production of many what is called thymic hormones, thymopoietin, thymosin, and many others. But they also directly interact with the T cells and are in part responsible for their maturation program and the process of positive and negative selection of these cells. Well, this is how it looks under higher magnification. So what we see, these are numerous lymphocytes. These are these ones with the darker, more compact nuclei, and we also see the bigger, like here, the nuclei of the epithelial cells. Some blood vessels are still present. So this is the general appearance of the cortex. What about the medulla? The medulla is, first of all, contains the very characteristic structures, which we call Hassall corpusculi. Hassall's corpusculi are the very good hallmark for the thymus. So during practical examination, when you have to discriminate between thymus and other organs, please look for this characteristic Hassall's corpusculi. If they are present, what you have under the microscope, this must be the thymus. So as you see, there is the number of the lymphocyte is quite smaller than in the cortex. So what we see is that there are many numerous blood vessels which can be seen here, and these vessels they serve for taking inside the immature T cells and sending to the periphery the major T cells. What happens to the thymus in time? It is that it functions till we became adult, and then later on it gradually disappears. So here you see such an adult thymus with involution. So you see that only very small parts display some characteristics of the part where the lymphocytes can proliferate. So in general, what to say, the organ disappears. So this is important, therefore, to stress that the education and production of functional T cells occurs usually in our childhood and younghood period. Later on, the possibilities of our immune system disappears. So this is why it is so important to be exposed to the different antigens at our young age, not at old age. At old age, usually the immune system is senescing, and therefore it acts not as efficiently as in younger individuals. Well, the very similar things about the function could be said when we are talking about the bone marrow. The only thing is that in the bone marrow, the cells which undergo the process of differentiation and elimination by selection and enhancement by selection, this will happen for B cells only. So let's switch to what we generally call the lymphoid system now. The lymphoid system is composed of a tissue fluid, lymphatic vessels, and lymph nodes. So what is the role? The role of the system is to drain the tissues and organs, and filter the putative antigens that may be found in these tissues and organs. The role of the lymphoid tissue is also to induce the immune response and trigger the immune effector cells, specifically activated immune effector cells proliferation. And finally, generation of the final response, which we are used to call, depending on the cells and the character of this response, cellular or humoral response. The cellular response usually occurs at the site of infection and is mediated by the cells which arrive by the blood, or resident cells, which may represent, if we make a comparison to the military, it is that they play a role of the infantry, which directly destroys the enemy. The human response, on the other hand, that is mediated mostly by the B cells. It is like an artillery, because B cells release their antibodies, which go with circulation, reach the site of a reaction. So the B cells may affect the enemy from a long distance. Cellular response usually requires direct activity against the antigen. Well, the lymphoid system is composed of a network of lymph nodes and lymphatic vessels, which is illustrated here. As I mentioned before, it covers the whole parts of our body except brain, the eye, and the muscular intestine. So all other tissues are drained by the lymphatic system. Well, the first important thing is where it is the beginning. The beginning is in of the lymphatic system is the tissue fluid. The tissue fluid is this is a fluid. One may say that it is very similar to the blood plasma. It is a blood plasma that is indeed which is filtered via the capillaries. And this fluid then, because it is usually present under some pressure, gets into the lymphatic capillaries and goes to the lymphatic vessels, the lymphatic ducts later on. So everything starts in the tissue, starts from the tissue fluid. This is shown how it works indeed. So we have a fluid there. Lymphatic capillaries, which are a little bit different from normal capillaries because they have closed ends, but they have clefts in the wall. So via these clefts, the fluid, which is under the pressure, goes inside. The pressure of the lymphatic fluid is, first of all, is responsible for the pressure of the lymph, which is circulating in the vessels. I don't want to focus too much on the lymphatic vessels. The only thing to say it is that they are very similar to the veins. I'm talking about the bigger lymphatic vessels. So they are almost. It is very hard to distinguish them from the vessels. the lymphatic vessels to distinguish from veins. Usually the capillaries, lymphatic capillaries are usually their lumen is very much higher, but as a matter of fact, it is indeed very difficult to distinguish them in normal histological slides. The capillaries they make a network. They are arranged into bigger vessels, and these vessels form a network, and in the eyes of this network we have lymph nodes. This is a typical structure of the lymph node. As you see, the lymph flows by few lymphatic vessels, and it goes out with one afferent lymphatic vessel. What is important that maybe this picture is not pretty well, because it shows that this part is as close, but it is not really true. The fluid goes inside and goes around the lymphatic tissue, which is arranged in the cortical part. We call it cortex. So as you see, and the cortex is occupied mostly by the B cells. So this is the place where the B cells are organized. Just below, in the paracortical area, we can find B cells. So this is the site for their activation and proliferation. And of course, we have also the medulla. And the medulla is this part where the serves for supply of the blood, of the lymphoid tissue with cells coming from the blood. And of course, it is also the place by which they leave the lymph node to go to the circulation. What is important to stress, it is that lymphatic cells usually do not get into the lymph node via lymphatic flow. They actively come with blood vessels. So the lymph node is colonized by the cells which are coming with the blood in general, of course. So this is a nice picture of a human lymph node. As you see, pretty nicely visible the cortical part and paracortical part beneath. And in the middle here, you see the medullary part. Well, this is a higher magnification. You see the capsule. The connective tissue of the capsule gets inside, as you see. So it may divide the territory of the lymph node into some smaller parts. We see the cortical part and paracortical part, and inside we see the medulla. As you see in the medulla, we see some aggregations of the lymphatic cells. These aggregations are around the blood vessels which supply the lymph node with the lymphocytes. Generally, the major organization unit of the cortical part is what we call lymphoid follicle. This is defined as a site for activation, proliferation, and maturation of B cells. This is a schematic presentation of this one. So it is composed of the so-called mantle, which contains densely packed, usually small lymphocytes, small B cells, and the inner part where the cells are activated, which we call a germinal center. And in this germinal center, we can distinguish first, B cells, which change into blast cells, Then blast cells, they change into cells which we call centroblasts, then centrocytes, and finally we get something you already know from the connective tissue class, plasma cells. These plasma cells are the cells which actively start to produce antibodies. We also, it is not like that this part of the organ is completely devoid of the T cells. Many T cells are present, but the role of these T cells here in the lymphatic follicle is to provide help and to regulate the B cell responses. So there are cells which are involved in activation of the B cell responses. What you see is there are two different pictures from the medulla part, where you can see the single plasma cells. They are stained with a pyronin, the agents which stains the endoplasmic reticulum, in particular ribosomes, in red. So we see they contain a lot of this stuff, and so they are very active. What is also of interest is, please look, here we have a blood vessel. Here another one, and these blood vessels are pretty nicely surrounded by a kind of the crown of the lymphatic cells. These are the cells which get in and get off of these vessels. What is also important to stress that the lymphatic organs, and including lymph nodes, contains multiple macrophages. These macrophages are responsible for uptake of some particles and presentation back to the T cells. So generally they act as the cells which are an important part of the filter, of which is a matter of fact a lymph node and other lymphatic organ. Well, so this is another beautiful picture of these vessels. In the lymph node, these vessels are characterized. They are a little bit different from normal capillaries. They are considered as being post-capillary vessels, which are characterized by high endothelial cells. High, it means that they are at least cuboid, or sometimes even higher than cuboid. This is why we call this special type of vessel high endothelial venules. These high endothelial venules are specialized in transmigration of the lymphoid cells from the lumen to the territory of the organ, and from the organ to the blood. Well, a few words what I need to say about the connective tissue, which forms a scaffold, which forms a stroma in the lymph nodes. This we call reticular connective tissue because it is composed of the type 3 collagen fibers which form this, what we call reticular fibers. Of special interest are fibroblasts, which are responsible for production of these collagen fibers. Why they are so important? Because they not only form a scaffold for the lymphatic cells, their role is far more advanced. As you can see on this picture, if we look at the arrangement of this tissue, we stain, for instance, specifically stain for the collagen fibers, we'll see that their arrangement is not random. We see that they go usually in some specific direction in parallel. So, as we see that from this, they usually go, they start at the subcapsular sinus, this place where the lymphatic fluid is getting in. So this prompted us to think that probably this arrangement is not really random, and indeed the further studies showed that these cells, they form a very specific arrangement. This is a cross-section of a bundle of the collagen type 3 fibers, as you see. This is a cross-sectional, this is a longitudinal section. As you see, they are all surrounded by the cytoplasm of this reticular fibroblast. So here you see a part of this. This is a dendritic cell. This is something different, but here is such a cross section of this bundle, and you see that this is surrounded very tightly by the fibroblasts, which form a kind of a tube. Indeed, if we perform an experiment where for instance, we add to the experimental animal some fluorescent dye, like Texas red, then we will see that this gets into the organ via lymphatic vessel, and then it is distributed along by this tube-like structures formed by these fibroblasts. So this can be seen here. You can find in the internet even some, movies showing how this goes inside. Nevertheless, what we know that the distribution of what is go to the lymph node is not random. It is distributed via specific type of tubes, channels, let's say, which are formed by the fibroblasts of the stroma, like you see on this picture. They indeed form a kind of a. It is not a vasculature, because the term of the vasculature is reserved to the cells of the blood vessels, of course. These are not blood vessels, but this is a system which distributes, a system of tubules which distributes the lymphatic fluid along the lymph node. The next organ is spleen. The spleen is composed in an apparently different way compared to the lymph node. It is composed of what we call white pulp and red pulp. What is the role of the spleen? The role of the spleen is identical to the lymph nodes, except because it participates in induction of the immune response. It participates in multiplication of immune effector cells. It participates in generation of cellular humoral responses. The only difference is that it, instead of lymphatic fluid and tissue fluid, it filters the blood. So the spleen is incorporated into the blood circulatory system. This is a short description how it looks like. So the spleen is supplied by the splenic artery, which forms branches. It goes inside, forming what is called the central artery, which next divides into the smaller, so penicillar-like arteries, which next go, and this is the very important thing, they go to the post-capillary vessels, which we call sinusoids. Sinusoids are characterized, as this is seen on this, by the very large diameter. And so they may play a part as a blood reservoir, as well as, the place where the particles may be easily captured by the macrophages, which are present in the wall of the sinusoid vessels. So here is generally the organization of the organ. What about this red and white pulp? What we call white pulp. These are the area which are occupied by leukocytes, whereas the red pulp, this is the area where we can find sinusoid vessels. So this is a similar picture which shows the same. This is how the circulation goes. We have a splenic arteriole which changes into a central arteriole. Around we have aggregation of the lymphatic tissue. In the lymphatic tissue, similarly as in the lymph nodes, we distinguish the part which is occupied mostly by B cells with formation of the follicles, as well as less organized T cell area. This is the small magnification of the spleen. What you see, this is a red pulp and white pulp. Of course, owing to the due to the staining, the white pulp appears to be red on this picture, whereas red pulp indeed is pale compared to the aggregations of the lymphatic tissue. It is red because, you know, this is the stain, the nuclei, and this area stain more with hematoxylin. What is apparent and what can be seen here, it is that all aggregation of the white pulp contains a big central arteriole inside. So everywhere you see here, this is the very important hallmark of this organ. So if you see only a small part of the spleen and you want to differentiate it with the lymph node, it will be not very difficult, because if you see that there is a blood vessel surrounded by the aggregation of the lymphatic tissue, this must be the spleen. Here is a part which shows how to distinguish. As you see, the white pulp full of leukocytes, mostly lymphocytes. Here you see the very nice follicle here inside the vessels, and outside more reddish because contains erythrocytes, the red pulp. Here the very similar picture again. The higher magnification of this area shows that, and here you can see that the diameter of this vessels is indeed very big. So this is to slow down the blood flow in this sinuses. You see along them, you see what we call splenic cords, the arrangement of the lymphocytes. And of course, what we can find here, these are very numerous macrophages. This one has been identified because it has obtaken some goods, so is easily visible. These are probably erythrocytes, because macrophages play a role of the cells which eliminate the old erythrocytes, which may be captured in the splenic sinuses. Well, here is a bigger view of the spleen. As you see, the follicle with a germinal center and the mantle around, and here around a reddish, the red pulp with sinusoid vessels. Another lymphatic follicle here. And of course, always what we see, these are central arterioles. Another picture, arteriole inside the vesicle, the follicle, and around some zone which probably may be occupied by the T cells. A few words about another type of cells which are present in the lymphatic tissue, which are very important. These are dendritic cells. The dendritic cells, they originate from the monocytes. The monocytes, when they reach the tissue coming out from the blood vessels, they change depending on the local environment into the cells of the macrophage-like type or dendritic cells. What is the role of the dendritic cells? The dendritic cells, they do not have the capacity to engulf, to phagocyze big particles, but they easily uptake a small one they capture them and entrap. And later on, they participate in the process which we call antigen presentation, antigen processing and presentation. And this antigen presentation is a phenomenon which is absolutely necessary for induction of a primary specific immune response. The most famous, let's say, dendritic cells, these are Langerhans cells that may be found in the skin and in the epidermis. Here you see the immunohistochemical reaction which shows them here, but their name comes from the very, very, sophisticated arrangement. They have many processes with many branches. This is, of course, everything to increase the surface of which will be able to expose the captured antigens. In the fluids, they become rounded, and they are usually called veiled cells because they have many processes which usually, when they flow, they are behind them. So sometimes it resembles the veil of a bride. Well, here I would like to show you two interesting pictures. Here you see the scanning electron microscopic picture of such cells. And look, this cell captured the antigens, the protein antigens, the molecules which are big enough, so they appear as a beads on these very thin processes of the cells here. Another interesting thing it is that under the picture done from the electron microscope, what you can see here it is that you see the surface of these cells, how it is complicated. This is also a very interesting thing because it shows that the antigen may be stored even for a long time on the surface of these dendritic cells. So the dendritic cells may also participate in what is generally called the immune memory. In this case, the immune memory depends on the storage of the antigen, which is very slowly released, very slowly presented to the cells, which results in let's say, the immune response, but at the very low level. Elicitation of this response is quite very easy, so the organism may respond pretty quickly. Our immune response may really respond very fast to the re-challenge of the antigen from the environment. Well, the cells which may be found in the T cell area are called interdigitating dendritic cells. You can see here, this is the body of the cells with a nucleus, and the T cells. Look how strong there is an attachment between these dendritic cells which present antigen to these T cells, to these T lymphocytes. Such type of interaction, very strong, we call immune synapse. So the synapses, synaptic communication is possible not only between the cells of the nervous system and the cells of the nervous system and muscle cells, but also in between other cell types, including immune cells. Well, just to summarize, as you see, what we have, this is we have a lymphatic system and the blood system. Lymphatic system operates on the basis of the lymph nodes, which are incorporated into and form a network within the lymphatic system. We have a blood system with the spleen. All systems are integrated. First of all, I mean, the lymphatic system starts from the tissue, from the tissue fluid, which then circulates by lymphatic system, is filtered by the lymph nodes, and finally goes back to the circulation by the thoracic duct, for instance. Okay? In the case of the blood is filtered by the spleen, which is incorporated into the circulatory system, And of course, it supplies, the blood supplies the tissues and lymphatic organs with the immune cells. So there is all systems are very well integrated. And at the end, I would like to mention also about the mucosa-associated lymphoid tissue, which is called mucosa-associated lymphoid tissue. It is present in many organs which contain a mucous membrane, like in alimentary tract, where we have a gut-associated, in the respiratory tract, where we, for instance, have a bronchially associated lymphoid tissue, and so on. You know that about some details about the systems you will learn, first of all, during the class on the respiratory tract and alimentary tract. So only very quickly I will go through this one. This tissue may be represented by single lymphoid follicles, single lymphoid aggregations, or these follicles may be associated, may form bigger structures, like, for instance, Peyer's patches, which are found in the jejunum. Well, the very interesting element of this system, which will be also shown in the class of this block, these are tonsils. The tonsils are present in our throat. They are, invaginations of our mucous membrane. And as you see, it is this mucous membrane is full of lymphatic tissue. These structures you see here, these are lymphatic follicles. As you can see, this is such a lymphatic follicle here with a germinal center and, of course, the mantle around. It is lined with an epithelium, squamous, stratified, non-keratinizing, usually epithelium. There are the invaginations which form what we call tonsillar crypts inside. And as you see, there is a lot of this lymphatic tissue here. As you see, the lymphatic lymphoid cells, they even, invade the epithelium. So you see the barrier between the epithelium, the border between the epithelium and the lymphatic tissue is hardly visible. This is another picture. As you see, there are usually many of these follicles here. So this is how to say our first defense system which is common for the respiratory and alimentary tract in our throat. Just only to mention about the lymphatic tissue I mentioned in the bronchial wall. As you see here, this is a bronchioles. Here, as you see, there is an aggregation of the lymphatic tissue. Similar structures we can find in the alimentary tract, as you see. Even we can see a follicle here. So as you see that the mucous membrane is infiltrated by lymphoid cells which form organized structures. And what is interesting, it is that the cells of the immune system may go to the epithelial tissue, so they may be present among the epithelial cells. We can identify them, these particular cells in the jejunum, staining them with specific antibodies against antibodies of the IgA class. IgA is a very typical type of the antibody called the secretory antibody, which is produced, which may be transported across the epithelial barriers to the lumen of the alimentary or respiratory tract. And here within the epithelium, we can see the B cells producing this particular part of the antibody. And just at the end, this is a summary of the mold. As you see, the mold is also integrated with the lymphatic system and the blood system. So this is everything for today. Now you will have a 5 minutes break before the next seminar starts. So thank you very much for your attention, and a message to my group. We will see with the first half of the group in a week on the next Monday. Thank you very much for your attention.
Excuse me, sir.
Yes?
Could you please just not forget to attach the presentation to the file?
Yes, of course, I will do it just in a moment. Okay.
All right, thank you.
Okay, you will find it on this